and start talking. So I'm going to start right now.
Okay. Welcome everyone, and welcome to CNL's Low Dose Radiation webinar series. My name is Allison and I'm with Canadian Nuclear Laboratories Corporate Communications team, and we're really pleased you can join us today. This series is proudly supported by Atomic Energy of Canada, Health Canada, and Canadian Nuclear Safety Commission. And before I turn it over to Edward Azam to introduce our presenter today, I would just like to draw your attention to the Q&A function at the top of your screen. We are certainly hopeful to have an opportunity for questions today, so you are welcome to enter your questions at any time throughout the presentation. And with that, I will turn it over to Edward Azam.
Thank you, Alison, and greetings to everyone. Today, it is my distinct honor to introduce Gail Wallace, a pillar and advocate at the highest level of the radiation research community's programs. Throughout her career, Gail made tremendous contributions to basic knowledge, radiation protection, and radiotherapy. Above all, she is a colleague and friend who is always there for many. Gail is professor of radiation oncology and radiology at Northwestern's Feinberg School of Medicine. She is Associate Dean for Graduate Students and Postdoctoral Affairs, Associate Director of her school's Radiation Oncology Residency Program, and its Centers of Excellence in Cancer Nanotechnology. She is also Professor of Bioethics at St. Vladimir's Orthodox Theological Seminary, Editor-in-Chief of the International Journal of Radiation Biology, member of numerous scientific advisory and editorial boards, author of over 200 publications, and editor of numerous books on radiation, cancer, and humanistic values. She is custodian of the largest tissue archive in the world, an asset to the entire radiation research community that is resurrecting the diligent inputs of pioneers in the field. Gail's contributions earned her the highest awards of the U.S. National Council on Radiation Protection, and notably the recognition of her peers and students with numerous awards for mentoring, whether at the high school level, graduate school, or residency programs. She is fellow of the American Society for Radiation Oncology and has been recognized early on by the American Cancer Society for her excellence in research. Gail's tireless service earned her many leadership roles, including chairing Subcommittee B of the National Academy of Sciences Committee on Low-Dose Radiation, a program that promises to generate new knowledge on low-dose radiation health effects, train a new generation of radiation professionals, and develop the necessary guidelines for protecting the public and workers. Gail's leadership roles at the NCRP, ICRP, NASA, NIH, ASTRO, and the RSNA are prominent and well-known. She does it all with grace and selflessness. Today, Gail will share with us findings and thoughts on low-dose and low-dose rate long-term animal studies. Thank you, Gail, for taking time to speak to us today. Thank you.
Ed, thank you so much. Thank you for inviting me. I'm grateful for this opportunity, and thank you for your very generous introduction. It's more than I expected, but thank you. I'm trying to get share my screen. It's sharing something. I'm going to stop share again because I think it's not sharing the right thing. Share this. Okay, can you see my screen?
Yes, looks good.
Okay, great, thank you. It doesn't look right to me, but as long as you can see it, that's what matters most. Okay, so what I'm going to do today is talk to you about long-term animal studies, particularly the low and the low dose rate components to those studies. I will say there's a heavy high dose rate and high dose component as well. And the reason why we focus so much on low dose and low dose rate over the years is because of the interest that there is. We know that we have a natural background and that we have human-made exposures that lead to a large number of exposures to our population. NCRP in 1987, the human-made component was quite low. By the 2015 report, it's actually making up about half of our exposures in the US. I think Canada is not so different from that. If you think about CT scans have only been growing in recent years. We can talk about whether that's a reasonable thing or not in a different conversation. Certainly there are many people that are worried about that, but it does seem to be of some concern anyway, that there are so many exposures. And then for occupational exposures, they're also increasing, and actually, in fact, they're decreasing. So what's happening for the occupational exposures is there was a big blip around 2004. Now in 2018, it's been going down, and that's been mostly because of protection, better protection methods, but also our awareness of the risks of radiation exposure, trying to mitigate them so that we don't have as many exposures in the population. So in the end, this has led, of course, to the standard controversy that we have in radiation biology. In epidemiology, we have a lot of data above the 100 millisievert range from patient populations, from occupational exposures, from many groups that have been exposed. The data are very reliable and give us a kind of good estimate of risk and its association. Below 100 millisieverts, excuse me, is where the data are controversial, and every kind of line you could ever dream about for what the data should look like, how we can extrapolate it, has been drawn. Some has been sublinear, some has been threshold, some has been superlinear, and it has been extremely difficult. In fact, there's some people that believe that a little bit of radiation is good for you. And so this is the controversy. So we've tried to look at this question to some extent, and we've also tried to look at the low dose rate question, which is also somewhat controversial. Sorry. Now, there have been large archival efforts done in the past, some with human data sets and some with animal data sets. These are mostly the human that have been well characterized. We all know the atomic bomb survivor data. We know about the Chernobyl data sets that have been developed. A large number of these have been found. What is probably less known and less studied are the animal exposure data. In my lab, we have the beagle dogs and the Janus mice. We have some other data sets that we've been gathering over time. The South Urals Biophysics Institute in Russia has their own animal systems and animal collections that they've done. Wake Forest has done quite a bit with animal exposures, particularly with primates. Fukushima has started to do their own data sets. There's the Frederica collection, which is mostly about ecological systems. And I just heard Chalk River is going to be doing theirs, and I'm quite excited about that. I think that'll be really wonderful. So in my opinion, it's really an imperative for us in radiobiology to generate archives and to study them, particularly because of these low dose and low dose rate effects. We now have statistics available to us that weren't available when many of these studies were originally done. So we can go back and look at the data sets, understand them differently, applying new statistical methods. We can put things like notebooks onto websites so that you can be able to have access to them, including metadata. I'm one of those people that believes that if you have the data set, but you don't have the other material that goes with it, then you have no way to check anything. So there are times when we've done archival material, we've looked at it, but then we've gone back to look at what we looked at the data sets, but we've gone back to the archive to see whether in fact the we agree with the diagnosis that was made initially, and we found some variations actually in what you would do today with new knowledge compared to what was reported when some of these data sets were first developed. So I believe that that sort of comparison is really important, and for that reason we maintain a tissue archive in addition to the data sets. So we worked a lot with the European Radiation Archive. They, I think, have a really a very good structure. The people that did a lot of that were Mandy Burschweltz, Paul Schofield, Bernd Grosch. They had a program from Melody to do this, and we actually were collaborators on this project, allowing for the generation of large data sets of material. We put all of our data sets and made them available on the ERA archive, and you can see the large numbers of animals that are currently available. They even do have some human data sets in with their data. They have much more in data sets than they have in available materials, but you can still get much of the materials. We have our own animal radiobiology archive that came pretty much from the DOE studies that were done between 1960 and 1995 in the US. We do have some other materials that have been added to the archive, and I will also say that I wish I could tell you we have every piece of data on the website. We don't. There are some that are still awaiting analysis and archiving. You know, it's just hard to find funding to put everything up, but right now we have most of the external beam animals for dogs, mice, and rats. I'm sorry, dogs and mice. The external beam rats we don't have up yet. We also have many of the internal emitters, particularly for dogs, that are up. Rat data we don't have up. We found some monkey data sets and some monkey slides that we are still sorting through. They look like they came from mixed exposures to radiation and other chemicals, but you know, the combined total is over 80,000 animals over the course of four decades worth of study. I don't think you're going to see anybody doing these large-scale animal experiments again. You know, 49,000 mice, all done collectively even though they were done in sequential order, done in the same facility with the same conditions. I don't think we're likely to see that. Same thing with 7,000 dogs at one facility, 7,000 dogs at another, 7 at another, all where the groups were meeting every few weeks to be able to discuss it. They're not likely to be repeated. So where most of our data sets come from and information comes from is from Argonne National Laboratory. No surprise, in my back door. I worked there for many years, so it was easy to be able to access that. I can tell you horror stories at the end of how difficult it was to get those tissues, but we did finally get them. We have some from Oak Ridge, some from Inhalation Toxicology Research Institute, some from UC Davis, Utah, Pacific Northwest National Lab, Colorado State, and Berkeley make up a smaller percentage of our irradiated archive. And as I said, we have pulled in some other data that were not necessarily part of those initial DOE studies. And you know, there are other groups. I was part of the Unscared 2021 report, and it talked about the difficulty of studying low doses of radiation and and particularly low dose rates of radiation, arguing that there's a need for more studies. And in fact, even studies that I'll talk about a little bit with DREF and where dose rate effectiveness factors, those are actually used when we're thinking about human protection. And there are, again, it's going to be archival data that's going to be important for us here. It's going to be hard to do the large scale animal studies that would be needed for us to assess what's going on at these very low doses. Here's a picture of what our paraffin blocks look like, and I need to say that most of our animals are preserved as paraffin blocks. It's remarkable what you can do with paraffin blocks today, but we don't have many frozen tissues. I know there are archives that maintain frozen tissues. The Department of Energy did not do that at the time. We have a wide range of doses for our mice, protracted low dose rate exposures, short-term higher dose rate exposures. And here's just an analysis of the relative age of the animal versus the exposure to either gamma rays or neutrons. Here's the neutron, here's the gamma ray. You can see we have, and then here are the controls. You can see we have a large, really a very large number of animals in these different groups. And of course, as the animals age, we get fewer and fewer. There were standard tissues that were preserved: lung, liver, spleen, kidney, heart, and anything that had a tumor or metastasis or anything like that. And the one thing I will say is that we do make these be available to people all over the world. So if you have an experiment you'd like to do, please just bug us and we'll be happy to try to send you some of the tissue samples. We don't like to send the full block out. We tend to send a couple shavings from the block, or maybe some mounted on slides, because the blocks are so important. So we've evaluated for, we've done evaluations for cancer incidence, for dose and dose protraction. Up to 50 gray of gamma rays was used. Dose rate effectiveness factor, we've done a lot of work using the so-called JANA database, which comes mostly from argon, where animals were exposed to neutrons in one part and gamma rays in the other to do a good comparison. We've published papers looking at DREF for up to 1.5 gray, DREF for up to 4 gray, and we have some ongoing studies that we haven't yet reported that I won't talk about, at least here. So this is a very busy slide, and I don't expect that you're going to analyze all this, but these were for 18 different experiments that were done. We took these from the ERA data set, which includes our own NEURA data set. And what you can see is these are Kaplan-Meier curves, different kinds of dose rates that were given at a given dose per, at a given survival curve. So this is like a survival curve for an animal. So in these Kaplan-Meier curves, you can see there are differences. There are some animals that died very young, at a very young age. There are some that died with a very kind of acute exposure. These differences are mostly due to age at exposure. One set are in utero exposures, one set are animals that were exposed when they were newborns. But in general, the curves look kind of similar for all of these, and we could then do these comparisons. We went up to 4 gray for our numbers so that we could get statistically significant numbers, and you can see these differences in dose rate that were evident, always with the animals that are untreated surviving the best. We did do a DDREF comparison, which is a dose and dose rate effectiveness factor comparison. What does this number do? This number allows us to see how we should modify our risk projections based on low dose rate. And what we did is we looked at the BEER7 model, looked at where our numbers actually were in 4 different large-scale experiments that compared protracted to acute dose. So what happened in BEER7 was they did a lot of studies where they took what was acute dose and they projected down what they expected it to be. We looked here only at experiments where there was an acute and a protracted exposure. In every case, the protracted exposure was somewhat protective. And these gave differences from the BEER7 report. The BEER7 is shown with the dotted lines. So we actually had differences that were very significant. We published this report a few years ago. More recently, and these are the data sets where we actually did different kind of analyses on what we believed. We've more recently done DREF instead of DDREF because DREF is more easily calculatable. We're looking not at the low dose effect, but at a dose rate effectiveness factor. So we're looking at the effect of a low dose rate exposure. What we got was actually sort of interesting. We found a DREF of 2.1, which means that the risk is about half at a low dose rate exposure compared to high dose rate. But if we compared the 0 to 4 sievert to the 0 to 3 sievert, the numbers are distinctly different, but also look at the error bars. Error bars are much larger for the 2.6. So one can argue that you should always go as low as you can, but statistical significance makes us want to go for a larger number of analyses. We did a lot of other kinds of analyses, and I want to point out one other thing that was very interesting. We had male mice mostly for one analysis for DREF, and we had female rats mostly. we got virtually the same DREF value for the male mice and the female rats, which suggests that, in fact, if rats and mice are pretty much the same, which they appear to be for most DREF studies, then in fact there's not a big male versus female difference in what the DREF should be for animals. These numbers are going to be appearing in the ICRP, International Commission on Radiation Protection, the report on DDREF that will be coming out, where the animal data are around that 2.0 range. We also compared, and then these are the female numbers with where we did the actual comparisons. Again, the control versus the protracted. You can see this very significant difference, which is very important. When we did the female rats, coming up with pretty much the same numbers as I just mentioned, and this says it's submitted, but actually was published about a month ago. Now, there's another interesting aspect of this. We've done this work in collaboration with Doug Borum and with Tony Brooks. So now I'm going to shift gears for just a minute for low-dose rate effects and look at dog data. We haven't done that much on external beam with the dog because most of the dog data were done with higher dose exposures. They did not have as many low dose rate exposures as we did for the mice. But what we do have are the animals that got exposed to internal emitters. And, you know, internal emitters give off different dose rates. Strontium-90, cerium-144, yttrium-91, yttrium-90. You can see here that what we're looking at is the dogs with lung cancer against the time, their days of life. And of course, the animals that are getting the highest exposures here that are at a very high dose rate are dying not of lung cancer, but they're dying of pulmonary injury. The rest are dying of lung cancer. Probably what's most interesting in these data are here, here for strontium-90, we don't see a big, we see the typical effect where the unexposed animals are giving lung cancer at the least rate compared to the exposed. Here with the cerium data, we see a flip-flopping where in some cases the controls are having more cancer than the exposed animals at the lowest dose rate. Same thing here for Y91, yttrium-91, and here for yttrium-90. It's clearly not 100% because we do see this difference with strontium, but what I think is important is there may be very big differences with internal emitters compared to external beam. And I get frustrated because a lot of the epidemiological data uses external beam and internal emitters like they're interchangeable. But if you think about an internal emitter, it goes in and it exposes multiple generations of cells to radiation, but normally at a very short distance. That's very different than what we get from external beam. There have been other studies that have looked at DREF, Tran and Little, using our data sets, published a paper in 2017, similar kinds of results. Doi in Japan worked on a different data set. They worked on a Japanese data set that I'll talk about in a few moments, and they too came up with pretty close to the same numbers. Braga at IES, who have been long-term friends of ours, they've done many studies with low dose and low dose rate, and Doi used some of their data when he did his calculations. As I said, there's a report that's going to be coming out in ICRP. It's still in its review phase, and should be available next year. That we'll talk about it. And Unskier did release this report, which is available actually on their website. Okay, so it's not just our data set. There are others in the world. I will tell you that Nature did up a small little article called Raiders of the Lost Archive. When our team went to Russia, looked at their data sets, looked at their data, and their animals and their tissue bank. We tried to have them put all their data on a website. Unfortunately, they never did it. I think it's just due to Russian laws about what they can make have people be exposed to, with how much data they can put out. But we were able to bring back a couple of samples with us, and we ran some of their samples alongside our samples with Trypan with H&E staining. We could see that we could get pretty good tissue exposure. This was an animal that got 5.5 gray versus 2 gray. And we then started to look at dog data sets. And in fact, there were a lot of dog data sets at Subi that we wanted to do a comparison with, but we just never were able to get access to it. We have our own data sets. This was done by Tom Fritz, who was one of the veterinarians that took care of these dogs early on. There's a dose rate against animals dying, and of course the acute exposures are here. And then we see the animals dying of leukemia, septicemia, and then many dying of tumors and even degenerative disease. These are from external beam exposures, not from any others. Now, also, so then the next thing we wanted to look at was not just total deaths of animals, but also cause of death associated with animals. And here is a Cox proportional hazards model. This was done by Alia Zander. She looked at survival probability, looking at all causes of death first, and then we're going to look at those from cancer next. These are the actual data. These are the data that are modeled for the Cox proportional hazard. And what you should notice is these are the untreated, the 0.1148 centigrade per minute animals here, 0.37, 0.67. So we've got these numbers. But what's kind of interesting here is that this green line corresponds to if an animal got roughly a dose of 1 to 2 CT scans a week. Now, I don't think most people are going to get 1 to 2 CT scans a week, but the fact is that the CT scans were showing a decrement here, which would suggest that in fact there could be an effect that you could observe. Now, she also did the neutron data sets here for total cause of death. And what's really interesting, again, the actual data, the model data, I want you to notice that most people argue that there's not much of a dose rate effect for neutrons, but in fact there is. And so these are animals that are getting the higher dose rates of neutron exposure. Not that much difference between these, but here at this very low dose rate, we do see a decrease we do see that the animals are surviving a little bit better, which does suggest that there's a dose rate effect for neutrons. We're not the only ones to report that. There are several people in the literature that have done it, that have reported that as well. But there might be some sort of like threshold for which there is a high LET dose rate effect that we need to put a lot more effort into understanding. We looked at lymphoma as a cause of death, so we're looking now at specific cause of death. This is the survival probability for lymphoma at different dose rates. This is the number of fractions that animals got survival at 3 different doses. And what we saw was that the higher the total dose, if the animal is given a fractionated response, it lives better. It lives longer prior to death from lymphoma. I guess that means that more fractions are more protection. That's not surprising. I think that's consistent in literature. These are the data for gamma rays. Here's the actual breakdown. When we look at the cumulative incidence function for these animals, this is the probability of dying of a particular kind of tumor. This is in the control animals. This is in the animals that were exposed, in this example, to gamma rays. A few things to note. First of all, of course, the exposed animals are dying sooner than the unexposed animals. But the other thing to note is that the causes of death are not also different. So what does that mean? That means radiation is enhancing the normal background rate of tumors, although there are some interesting exceptions. So one of the questions that NASA has been asking for a long time is, there's an increased incidence of radiation-induced lung cancer in Japanese atomic bomb survivor women compared to men. Does this pan out for animals? At least for mice, the male to female differences show a higher risk for males than for females. So this would suggest that that would make a very big difference, that either the mouse is not a good model for the human, or maybe we need to rethink the Japanese atomic bomb survivor data. Now, I promised to mention the Japanese work. The Japanese have a facility. They have an experimental research facility up in Aomori in the northern part of the country. And Ignacia Braga did a lot of this work. These are her animals, and these are our animals. We probably don't have a comparison for the 0.4 gray exposed, but we do, and we have the 0.43 gray, so we have the lowest dose, but they gave theirs at a low dose rate. We gave ours in 1 fraction. We have the sham fractionated, they have the sham fractionated. They have the 7 gray, we have the 7.74 gray, and I guess their animals were done in the past like 5 years. Most of the animals we were looking at were done, they ended in the 1990s. A few things are really important to say about this. Number one, that most of the causes of death of these animals are the same. What are the exceptions? Respiratory system and digestive system. These are the data on the female animals. The next slide here, I'm showing you the male animals. Again, respiratory, digestive system are most of the differences. We believe that might just be due to circulation, since their circulation facilities are really different than these were, and due to bedding, since their animals were germ-free and ours weren't. So I think that's really interesting. Another point that I think is important to make for our animals in particular, I haven't seen those data coming from the Japanese, is that the sham irradiated animals at particular dose rates, we actually couldn't do the comparisons between the unexposed and the exposed, because the unexposed sham irradiated that were taken to the chamber like 400 times, they had a higher cancer incidence than the 400 sham irradiated that were not taken to the chamber. And that suggests, I'm sorry, let me say that again. 400 sham irradiated taken to the chamber 400 times, and the unexposed controls that were sitting in the room, age matched, strain matched, everything matched, they had a higher incidence. So just the stress of taking the animals to the irradiation chamber caused them to have a higher cancer incidence. We're trying to get the Japanese to look into that for their data sets as well. Okay, let me shift almost more exclusively to the dog data now. I mentioned some dog data earlier. We found about 5,000 dogs that we could do easily for external beam. These are in our own archive. They were done at Argonne, 50,000 tissues, normal tissues, tumors, metastases, and the standard normal tissues were lung, liver, spleen, kidney, heart. So all of those are available, again, stored. This was the original irradiation chamber. And here you can see the dog phantom that was used for these studies. Now, initially, there was some published information, but honestly, it wasn't as much as I would have thought would be appropriate for the circumstances. They had they did some stuff on life shortening, tumor induction, tumor sensitivity, probably a lot more on the leukemias than on the solid tumors. Male versus female differences were brought out, age specific differences, types of tumor differences. We had done some selective molecular biology on the tumors, and some stuff, and particularly a lot was done by Tom Seed's group on hematologic processes with these animals. Here's a figure that Tom Fritz, one of the early veterinarians, had done, and what he was looking at was the percent mortality from different causes and the time to death. The animals that were dying in early death were getting a very high dose rate. They were dying mostly of other causes of death, septicemia, aplasia. As we get older and older animals, the cause of death now became other causes and fatal tumors. So you can see this distinction with the different dose rates of exposure. The lower the dose rate, the longer the animal lived, of course, but also the cause of death became quite different. And this again says pretty much the same thing, where these animals are dying of myelodysplastic syndrome and leukemia here, and they're dying acutely of anemia and septicemia. Bruce Carnes was working with these data sets years ago, and one of the things that he did is looked at the cause of death, but he put together the beagle dog, the mouse, and then extrapolated what the human would look like with these error bars. He made the lifespans be contracted so that it looked like a mouse and a dog would look he used the data from the beagle. He put in the mouse. Sorry, put in the mouse comparison, and then extrapolated what humans would look like based on that. And of course, lower the dose rate, the longer the survival. Higher the dose rate, the shorter the survival. Now, we have, from our internal emitter dogs, there were about 7,000 dogs per site. They were not done at Argonne. They were done at Lovelace by inhalation, by University of California, Davis, Utah, and Pacific Northwest National Lab. They were both injected and or ingestion. Argonne did the external beam studies. There were 7,000 dogs per site. They maintained these dogs for their entire life, and nobody expected it, but some of the dogs lived to be 23 years of age. They just took really good care of them. And I mean, it was the low dose rate animals and the untreated that lived that long. There were early toxicities. They looked at cancer. They looked at life shortening. And this was an early slide where they looked at the radium. They had actually looked at mammary tumors in beagles that had been given radium. And you can actually see the radium causing these little star patterns inside the beagle's mammary gland. This was a non-pregnant female dog. So this was an internal emitter. They had evidence for it, and they could find it in sites other than where they had exposed the animal. Now, when we did this again in collaboration with Doug Borum, when we looked at the different radionuclides and what the total dose was that was delivered versus the age at death, the different radionuclides caused a shorter survival time based on the kind of dose rate that they gave out. So again, demonstrating that these internal emitters could be used to study dose rate effect. When we looked at the cause of death, the lung cancer was the older animals. Pulmonary injury was for the younger animals. And when we looked at the decay versus the cell cycle, this is going to be important. As there's decay, we could see that the average dose per cell versus turnover went down. So this is the difference between the internal emitter and the external beam, is that there is this exposure that can take place. And when we look at the cell turnover per generation, comparing the percent survival against the days, all animals were the same age at the time they were exposed, but the different radionuclides had different effects and different dose rates and caused a different dose per cell turnover. And we believe that that's probably one of the reasons that there's a big difference in the dose rate effect for these different radionuclides. Let me conclude this and go on to some of our more recent data. So in addition to the international data, tissue sample archives, we use these archives to look for more new information that can be done differently than it has been. We also hope to improve international collaboration. And for the animals exposed to the external beam and radiation models, we could do new kinds of statistical analyses that we couldn't do otherwise. We saw similar results from animals that were exposed to the Japanese and the Argonne studies. And as I said, taking a large number of animals to the chamber caused exposures that were different. Now, we've been doing some different kind of analyses of the animals that are internal emitters. And I'm going to show you first of all, some data that maybe we generated 2 months ago, and then I'm going to show you some data that we generated like 3 days ago that I'm super excited about. So I'm sorry you're the first audience to hear this, but I was so excited I couldn't hold myself. This is a lymph node from a dog. The dog had inhaled silicon microbeads that went into the lungs, got trapped in the lungs, but also probably traveled elsewhere in the body. We're showing here that it's in the lymph node. What are we doing here? We have an instrument at Argonne National Laboratory where we can actually look at specific elements on the periodic table, and you wouldn't normally find silicon. We don't normally find silicon in the lymph node of animals. Here's the dog, here's the silicon. These are the silicon microbeads that were put into the animals. Here's the H&E stain, so we can actually see the lymph node, and we can look at where these sites are. Inside the silicon microbeads or yttrium. The yttrium has decayed by now. We can't really look at it. We can quantitate how much silicon we have. We can hopefully very soon be able to tell you how many atoms of silicon are actually present in this sample. But what I've been excited about is the ability to not just look at the silicon, but to look at the daughter products. So it's true, there's no, so I'm sorry, these animals got strontium, not yttrium. I was thinking about the daughter product. There's almost no strontium left. There's almost no yttrium left. It's all going to be zirconium. David Hooper did for us this decay to calculate what we would expect after all these years of these beads living inside the animals, and we would expect that it's going to be zirconium 90 that is inside these animals. And looky, we're seeing zirconium that lines up with where the silicon is. I know this doesn't look like a lot, but in fact, when you quantitate it, this ends up being quite high. We also see iron in these same locations, which we think might, we have to relook at the beads carefully, but we think it might be macrophage. Macrophage usually have a very high iron signature. So we think maybe these silicon beads were taken up by macrophage. And here is a blown up image. This is the silicon again, where we're doing the overview scan with the H&E stain, where we're actually getting in at finer detail to be able to look at this. And here is the silicon and the iron, which we believe is showing us where the macrophage might be in these particular samples. So I'm really excited that now it looks like we're going to be able to look at many of these samples. We've done a larger number of dogs now. I have a student, Tony, who's been doing a lot of this. I'll mention her name in a moment, but this has been very, very important for us. So we actually believe that these animal archives and the old animal models can help us to look at things like DNA damage and the damage response. And we can go back into these old tissues, look at what's going on there for macrophage immune responses and other sorts of changes, and then do new animals to test that model. So we develop a model, we look at it in the large scale animal studies where we can do, you know, hundreds of animals, and then we go back to a few smaller animals to be able to compare it. And we support the idea of being able to take these models and these data and model them even better with time, where we think, especially for the internal emitters, cell cycle time is going to be important. How long they've lived with those exposures is going to be important. Effects on different kinds of components to the tissues is going to be important. And I need to say that, like, while I showed you the data on strontium, we've done some with yttrium. Plutonium is going to be important, and these have different ways in which they expose. Like plutonium, for instance, we have some other published data that argues that plutonium doesn't go to the nucleus. So we're not getting as much DNA damage from that radiation as we're getting cellular damage. So what's going on in organelles there is going to be very important to be able to draw that conclusion. So my final conclusions are, look, large-scale animal studies, we can learn a lot from them. We need to continue to do them. And I believe combining data sets, we can even do more. So I'm hopeful that Chalk River will want to collaborate with us so we can combine our data sets to answer even larger questions than we are now. I think these worldwide efforts need to go on. And I've been a proponent for trying to form an international consortium to get together to talk about databases, archiving, and the information that we can do, and designing experiments that are very large. The things that we could do with combining our data with the ERA data sets were much greater than we could do alone. The comparisons between our data sets and Japan were very, very important for us. And I do think new modeling approaches are going to be extremely helpful here. This is our group that's doing mostly internal emitter work, and includes Wes Bolch from Florida, Rebecca Abergel, who's doing a lot of the new animal work with mice from Berkeley, Shaheen Duji, from Georgia, who's doing Georgia Tech, who is doing a lot of the pharmacokinetics with us, and then we have David Hooper, who I already mentioned, it helped us with the half-life of these animals, and then Ashley Golden, who does the statistics, and then other components, other people who are playing key roles in this work. Tanya Paunescu from my own group, who does almost all of the imaging by X-ray fluorescence. From my own team, I especially need to mention the work of Tony Copeland-Hardin. Tony is the graduate student who did a lot of those images that I just showed you. And Alia Zander is a graduate student who has left the lab. She did the cancer incidence in the data sets, and Ben Haley, who did the DREF studies. And with that, I'll end, and I'm happy to take questions.
Wow. Thanks, Dr. Wallace. We do have some questions.
Yeah, I need to get off of my share screen somehow. It'll take me, there we go. I think I can do it this way. Okay, ready for questions.
Okay, so I'm going to start with this one, actually from Dr. Brooks. With the new EPI studies, molecular biology and animal studies, is it time for a new beer 7?
Thanks, Tony, for that question. People have asked that question for whether it's ready for beer 8. I agree. I think it probably is time. I mean, I think there's enough new data out there that and there are errors, and I don't think that there are errors. I think that there are stories that are incomplete in beer 7. For instance, we disagree with some of the conclusions in beer 7, but honestly, they didn't have access to the same data sets that we have now. So I think it's time to think about doing a new beer 8. I would be very supportive of it.
Okay, next question. Can you talk about the possible mechanisms of higher cancer incidence in females than males?
Yeah, the possible mechanisms. I'm not 1000% convinced that I believe it. Okay, because of that Japanese atomic bomb survivor study, but what I guess people should look at is the timing of when that incidence is different in females versus males, and can you relate it in any way to hormones? That would be one thing I think that should be considered. When people have done like dose calculations, it doesn't look like the dosing is all that different from male to female for just a routine external beam exposure. So I don't know. The hormones are probably the only thing I can think of that makes some sense.
Okay, so next one, how the DREF is calculated from external beam gamma radiation and internal beta radiation would be, I guess, how you would compare those.
For internal emitter versus external beam, yeah, that's a really good question, and I don't think there's a good way to do it, and that's the problem. The internal emitters, you have to take into account that you're having this kind of continuous exposure, so you need to take into effect the dose rate, which is going down with time, as opposed to the external beam. So you have the difference is you have these few cells that are being exposed. Even if it's a beta emitter, you have a few cells that are being exposed, or a relatively larger number, and versus an external beam where you're having many more cells exposed. So I think we need to think more about what's the biology of what's going on for the internal versus the external. We have never tried to calculate DREF from an internal emitter. Maybe it's time for us to think about it, but we haven't thought about it yet. So it's a very good question. Thank you.
Okay. So next question, there are many laboratories around the world, like the Chalk River Laboratories, where the data on the radiation doses received by the workers are available. Has a comparison been made on the overall health of these workers with the overall health of non-radiation workers in different industries?
I don't know about the Chalk River data sets and whether people have looked at them. I just don't know. But I know that there are a large number of studies, like for instance, in the US, there is still the 1 million person study that's ongoing, where it compares radiation workers, mostly from like the 1950s, 60s, 70s, where the doses were high. If you were to compare today, you wouldn't see much of a difference because radiation protection is so very effective that people aren't being exposed. I think I kind of showed that in the first few slides. But in the early days, when things were not so protective, you can start to see some differences. I know that there have been a number of efforts done internationally to compare, and certainly some of the Canadians have been involved in those studies, but I don't know whether they've included the Chalk River kinds of studies. Again, I'll say I think they have to work with early exposures, not with exposures that are much more current than they are now. But I think it would be, if they haven't done Chalk River, they should be done, and now with the large consortia that are working together internationally, that would be very important.
So the next question is definitely multi-pronged, so bear with me here. I'll publish it in a second. Is there other evidence or studies that have also demonstrated that shama radiation alone increased cancer rates?
Wait, can we just stop and I'll answer one at a time?
Yeah, go for it.
I'm never going to remember.
Yeah, okay.
There is some evidence. There is some evidence, but I don't think there are that many studies. We don't see it in our own studies for the ones that have gone to the irradiator, 20 times. So I think that there's a given amount of stress before it gets to be so significant that you see it. And I don't think there are that many studies that have actually looked at that many shama radiations. Now, the only ones that maybe have would be Japan, and I've asked them to look at it. They hadn't been looking at it. I've asked them to look at it. We'll see what they say.
So building on that, does this mean that studies theoretically could have been attributing adverse effects to radiation rather than stress or other confounding variables?
I think that's always the fear, and that's why it's really critical to do the shama radiations all the time. I mean, if you're always comparing shama radiated to real irradiated, then you shouldn't have a risk of having a problem. But if you're not comparing, if you're not comparing apples to apples, you're going to get a different result. So, and most people are pretty careful nowadays, and they do the same number of shama radiations as they do regular radiations.
So last part, if so, would you argue that animal studies should use two controls, shama radiated and a pure control, as a standard for radiation biology studies?
That's the only way they found this. Okay, so they were so careful with these studies. They used to rotate the animals in the animal house every day because they were afraid that there might be a particular part of the room that had an effect on the animals, so they can minimize those effects. They were really very careful about how they did them. They believed that sham irradiation to non-irradiated control was important, and indeed the data bore that out. So I think there is a reason to try to do non-irradiated, just sits in the chamber, happy little campers, nothing going on in their lives, and they do seem to have a lower incidence of cancer than many of the others. So I think I think it's worthy to do it that way.
Okay, next question. Excuse me. Your explanation for the difference in biological mechanism between external and internal radiation was very helpful. Do you expect to see similar differences between different types of internal emitters, alpha, beta, gamma?
Yeah, I expect that we are going to see differences because alphas are, something very, very different than betas do. The alpha has a very short range, but it's much more damaging. So I expect that we'll see differences, but data have often surprised me, and until the data come, I'll wait. Thank you for the question.
Okay, so Paul asks, hi Gail, what's your opinion on using AI and machine learning for low dose radiation tissue studies given subtlety of morphological changes?
Yeah, we've been using, I didn't emphasize it, but a lot of our imaging with X-ray microscopy, we've been trying to get the AI to work with it to help us locate things a little bit more easily. Right now, we have to hand locate all our or eyeball locate all of our silicon, zirconium, all those things. Now, if we could have an AI that can identify where those are first, so that when we know where to hone in on it, that would be important. And our Argonne actually has a big computer group that is trying to do that, with us. I also think that with large data sets, we did work a lot with machine learning for some of the analyses that we did. I didn't show you everything. I don't think we could have done as much as we did without machine learning. I mean, in many cases, we were comparing 230,000 mice with, 30, 40 different parameters. So you're seeing kind of the synthesis, but it was much more complicated than it seems. And I think as you get more and more data sets, you have to go with at least a machine learning approach to much of it. So I strongly support that. And the other thing that is a worry, it's not really an AI issue, but we generate for every pixel on those microscopy images that I show you, we have data on every element of the periodic table. So that's why we can do silicon one day and then go back and do a zirconium analysis without having to do a new scan. Well, the thing is that it's a huge amount of data that we're collecting, and so if we didn't have like big computers out of Argonne available to us, it would be really hard for us to do a lot of those analyses. So there's also something to be said for these large computers too, not just the AI and the machine learning.
Thank you. So Gabriel noticed at the beginning of your presentation, he saw Hormesis. I hope I'm pronouncing that correctly. He's just hoping to understand or hear you talk about that and its relevance to low dose exposures.
Okay, so I just taught this in a lecture this morning, so I'm kind of all tuned to it. So Hormesis is sort of the idea that a little bit of radiation is good for you. You should go sit in a radioactive spa, lap up those rays. You're going to get, everything's going to be good. And we do have data that support hormesis. I mean, we can show some stimulated immune response, and what the heck, those animals that are exposed to radiation and getting a lower risk of cancer, I mean, that is a typical hormetic response. I don't like to use that word because it has kind of a bad connotation in the literature. I would prefer to think of it as some sort of like adaptation or something like that. The problem is that we can't figure out how to drive it. So that's why we need to study it more. If we could be exposed, how do we know between those two animal groups whether you're going to get the increased risk of cancer versus the decreased risk of cancer in a given situation? And how do you know in any individual animal which one is going to happen? If we understood it and could predict it better, it would make for a difference. I do think we see it for internal exposures. We've certainly seen stimulation of immune response with external beam, but we haven't seen that sort of like protective behavior with cancer with external beam. I'm not trying to say it doesn't exist. I'm just trying to say there's a lot more work to be done, and I do worry about the negative effects that also exist at the same time that you're getting hormesis. We need to understand it better in order to figure out how we can dissect those apart. I hope that answered.
Okay, great. Okay, I think we can squeeze in a final one here. So how far have molecular analyses, examples, regulation of DNA damage response genes, and using archive material progressed?
Yeah, so from our end of it, there are lots of things we can do. We've published back in 2000s using archival tissue for these archival tissues for DNA sequencing, and we did quite a bit. We could do quite a bit. There are new methods now to be able to get entire genome sequencing done from archival samples. We've worked with a few people to do that. What I do think is you have to pick your samples very, very carefully. When you're working with these large animal archives, sometimes animals died, 6 hours before. They were kind of moribund when they went to take the tissues, and those are not going to be a good choice to do for these to use for DNA analysis. The DNA is already degraded, but we've been working with a company. We're trying to get some sequence data. We'll see how that turns out. I can't tell you any results with that. It does look like different methods of preparation are better. Some are better than others. We certainly can get microRNAs out of these things. We have a paper that we've been sitting on, and I've been sitting on, it's my fault, on microRNAs from mouse data sets. Kind of waiting for a picture to come out a little bit better, but the data are consistent. They look really, really pretty good, so we'll probably publish that soon. And we're working on that with dogs right now. I'm pretty convinced that we're going to be fine with microRNAs. We can certainly do some immunohistopathology on these samples without too much trouble either. Can we do mass spec for protein analysis? Can we do messenger RNA? Messenger RNA, I just don't think we're going to be able to do it. There's just almost nothing there to be able to look at. Even with modern methods, I think we're going to have problems. Mass spec, the problem is the proteins are cross-linked, and that makes for some difficulties, but newer methods are coming out. We might be able to get some data out of that. I was just hearing how in Japan, there are people that are doing it from our fixed tissues. They're able to get mass spec data. So, we're going to try. So the answer is yes, there are some, but I think it's limited, at least yet still, to what we can do.
Thanks so much, Gail, for an amazing seminar. We are really, really grateful for the time you have taken today. And thank you all for participating. This is our last webinar of the year. We wish you all a wonderful holiday season, and we'll meet again in January of next year. Thank you. Thank you, Gail.
And thank you, Ed. This was great. And if anybody has any questions or wants to get tissues, please feel free to give them my email address and have them contact me. And happy to talk with the Chalk River people too.
Absolutely. Thank you, Gail. Thank you. And this will end our webinar for today. Thank you. Thank you.